Okay, so good morning, everyone. I was asked to give some concluding remarks on the series of talks that were given on the subjects of crosstalk and stress and the use of priming technologies as a way of ameliorating these stressors. First, I wanted to say that I appreciate your attendance and the interest you showed in the subject, although I think it is a timely subject, so it makes sense for young researchers to actually be interested in related research, in related activities. It was an intensive course. I realized it was quite demanding, but hopefully you have gotten some basic information that span from more introductory concepts, such as terminology, the background information regarding stress responses in plants, then moving on to deeper insight about molecular mechanisms and pathways that are actually followed during such responses. Of course, having an in-depth discussion about signal molecules that are involved, because these are effectively also ideal candidates often to be used as priming agents for relevant technologies. And then, of course, highlighting the various subcategories of priming technologies, which do include chemical approaches, biological ones, as well as nanomaterial ones, which do actually represent also movement forward as the next generation priming using chemical molecules. So I think it is undisputable that this is a time of change, climate change. We are all affected somehow, one way or another, from some sort of stressor. Perhaps the reality here is different to the reality it is in Cyprus or Greece or Peru or what have you, but you can rest assured that a farmer in Peru will have equal difficulties securing sufficient yields of the crop of interest in that region based on their geoclimatic conditions in comparison with the farmer here in India. Priming does offer a wonderful solution, I believe. It does offer extreme versatility when it comes to the means of application. As we saw extensively, there's many ways you could actually tackle this issue: through foliar applications, through soil watering, through seed application. So there is flexibility on the part of the scientist, of the researcher, which is great because it gives us lots of space to further improve our approaches. And of course, doing this in a speedy manner in comparison, for example, with the conventional breeding approaches that have been used for ages now in agriculture, but at the same time also representing a non-GM, green, sustainable approach that is quite important, especially for regions that are not as receptive to gene editing technologies. Just this morning I read an article that recently the EU Parliament was asked, or various ministers of EU nations were asked to reconsider the notion of having gene-edited crops as GM ones, and apparently skepticism remains in the EU, which has caused a lot of concern in the field of biotechnology, because of course it is widely accepted as a technology globally, and certain regions do seem to be falling behind. But in any case, priming actually does offer a wonderful solution regardless of such approaches, and at the same time these approaches are not exclusive, mutually exclusive. So as we've seen, in the future priming approaches could indeed be combined with gene editing approaches, for example, so as to create the next generation of stress management principles that would lead to indeed much more improved yields. In general, I mean, this is the main comments I wanted to make, and it is important for me, however, to also hopefully get some feedback from the attendees. You represent the young generation of Indian researchers. Of course, you're in good hands. the academic profile of your tutors is outstanding, but you represent the hope of Indian research in the future. And of course, my advice to you as an older researcher is to always try to remain innovative, never stop thinking, never stop reading. As I always joke, now I read more papers than I did when I was a PhD student, which is true, But it is actually the only way to stay on the top of your game and to continuously seek for new ideas, always with the intention of actually trying to find solutions for the end user, which is the grower, the regular farmer. So inquisitiveness is important. Don't be afraid, as I tell my students in my group, don't be afraid to try things. Don't be afraid to test things. At the worst scenario, it will not work, but this is biology. I mean, in biological research, things don't always work. And even if they do work, often you have to repeat several times and tweak and optimize until you actually find the optimal solution. So try. And this is also in relation with some brief questions I got in the previous days, because there are no easy answers. There are no easy solutions. It is all about getting a proper experimental setup arranged, because this is the most important thing, and then just trying. So as I said, I mean, it would be wonderful if I got your feedback, any sort of feedback in terms of both the content of the lectures, but also even specifics, if you have specific technical even questions or any other form of interaction you would like to have. So, by the way, I see we have our friend back again, the famous cat. Every day we've been having a cat that is attending, so ready for the exams as well, I think. That's very good, very good. So,
yes.
Yes.
So I just wrote on the group that
Yes.
we have created, WhatsApp group.
Yes.
You are making concluding remarks. So is there any question?
Yes.
I can get your insight. So there is one question. What are the possible approaches to dodge plant defense mechanisms for delivery of secondary metabolite tagged nanoparticles?
When you say dodge,
Evade.
Evade responses, why would you like to evade responses? I mean,
I don't know.
that doesn't really...
yes, it doesn't.
Yeah. Well, in any case, the field of delivering secondary metabolites, actually, this has kind of been covered, because secondary metabolites, this actually includes natural metabolites. God knows I've been talking about the use of melatonin, the use of hormones, which are effectively secondary metabolites, you know. So I have spent the whole lecture discussing about the values of actually functionalized materials with chemical priming agents, including plant secondary metabolites. So the one thing, though, that is differentiating itself is what I mentioned yesterday, I think, that in addition to directly applying single molecules, secondary metabolites, of course, nowadays there is a big movement of trying to really take advantage of whole plant extracts. It is extremely common, as I said. I mean, we're also involved in a project. Actually, yesterday there was the closing meeting of the specific project in Cyprus, but of course I was here, but well represented by the people from my group, where, if you remember, we actually surveyed, for example, the local flora, basically identifying local medicinal aromatic plants of Cypriot geography that have been shown ethnopharmacologically through word of mouth, through practical empirical use from elders, to actually carry out extractions with different protocols from different organs in different periods of the year to see if this could be used indeed as a priming approach. And I'm very pleased to say that there have been some data that are extremely interesting, because this could easily also be employed in the pharmaceutical industry and so on. So yes, it is possible to actually carry out nanopriming, functionalizing with secondary metabolites, but actually are using extracts and not even single molecules. The only comment I will make in that is that using extracts or cocktails of microorganisms or any sort of priming solution that is mixed, the only difficulty, perplexity, is that it is just more demanding to pinpoint the mode of action. Whereas, of course, using single molecules or single strains of microorganisms, of course, it is relatively more straightforward to actually try to go to deeper mechanistics about the way it works. So, yeah.
There was one more question yesterday, but we could not.
Okay.
Yes. The question is that we have developed various nano formulations as far as agriculture sector is concerned, and in trials, agricultural trials, in field trials, they show effect.
Yes.
Right. But when the farmers apply these nano formulations, the effect is not so visible.
Well,
what may be the possibility?
right.
And sometimes they show effect, sometimes.
I see. Right. As you are all aware, reproducibility is a major element that is required in a successful technology. It does happen that you get variations, but this is also part of working with living organisms. Of course, at the end of the day, an application in the field would also be relevant to exogenous climatic conditions. There is so many factors that could go wrong, but in this situation, I also need to stress out the importance of proper practices. Farmers also need to be well aware of the proper practices. I am not suggesting that these practices are not followed, but of course, the first thing we always say when something goes wrong in the lab, for example, is let's see if there was some sort of operator error. So it is also important to train farmers so as to properly handle such material, especially if they involve the preparation and handling of the material pre-application. So my only question there would be whether this is seen routinely for the farmers to not be able to reproduce this, or it is operator specific or what have you. So many factors could go wrong, but actually one of them that you should consider is proper training of the personnel that actually do carry out these procedures. Because if you're careful, actually chances are that things will go better than if you don't really follow the standard practices the best way possible. Yeah. Everybody makes mistakes, of course, but it is imperative that full attention is paid when you carry out experimentation, of course. Any sort of comment, any sort of question?
Dear sir, I personally want to thank you from the bottom of my heart for delivering such an intellectual lecture. Previously, I was not known to this topic too much, but after hearing you, I have developed interest in this topic, and I want to incorporate this in my research.
Oh, that's wonderful. That's wonderful. This was the intention. The intention was to actually, hopefully, gain your attention and see the benefits of this technology.
My work is on diatom, so I was not having prior knowledge of this topic. So now I wish to incorporate this.
Wonderful. Well, I'm happy to hear that. Thank you. Thank you so much.
Sure.
Yes.
So, sir, I've been a participant here. So, as Geeta said, she relates herself to diatom. So, I relate myself to the animal biology. So, this process was actually about the seed priming, but I correlate it as my priming towards plant stress plants as well as plant stress.
Yes.
Because all these terms were, we studied way back, but now it is just a refreshing and revising things. And more of it, I could also correlate it in the meantime, to all the new technologies that we use with animal biology.
Of course.
Like chromatin modifications, DNA, RNA, signal molecules, like everything. So for me, it was very engaging. And whenever I used to think of a question, I could find it in the next slide, the answer.
So this hearing from most of the students. My
Okay.
PhD scholars.
Okay, that's good. That's good. That's good. That's good.
The
How-
slides you prepared were so exhaustive.
Too exhaustive. I'm sorry about that.
No, not that.
But
We're not. You should not be sorry. We are congratulating you for that
Yeah.
and appreciating you for that. That you took the time to prepare such a detailed description of everything. So we thank you, sir, for that such informative and engaging lectures.
Thank you.
And welcome you again to our sessions and to India as well.
definitely,
And
definitely.
welcome back again.
Well, the ties are strong with also collaborators here, so of course this is not the end of it. But this is the interesting thing, though, that this is an approach that could be horizontally applied. The first thing that, when I first talked about the concept of priming, it was you actually who did the very spot-on comment that this actually sounds like an analogy of vaccination for the animal kingdom. So there are many common elements across different organisms, of course. And also, as you saw, this is also represented from the variety of agents that could be employed, such as my beloved noshaspirin that was developed for human use, for example. So things, again, are not mutually exclusive. And this is another reason why I ask you to remain inquisitive. For example, I came up with the idea, although this was material that was published in research relevant to cancer biology. So actually having kind of a more narrow mind and saying, okay, I'm going to use only specific keywords and look at specific journals, and that's it. You actually miss out on wonderful opportunities. And I think this is a very good example. Because if you kind of think outside the box, this is when often you will get the really promising ideas.
So apart from the lecture, we have also learned punctuality and dedication from you. So you are so punctual.
There is no other way. There is no other way. I mean, as I always tell to my children in the lab, fun is fun. I also like enjoying ourselves. We have group gatherings. We will go out for lunch together. So fun is fun, but work is work. It's completely different things, you know. And at the end of the day, professionalism at the workplace is imperative. And if this is covered, then fun can also follow and should follow. Because at the end of the day, you're also students, you're young people, you're also supposed to enjoy yourselves and have fun. You're not supposed to just be a bunch of robots that are like in the lab 24/7 and of course not. You know, this is also part of having fun and enjoying your everyday reality. But professionalism is of course a given, so excellent. No, I was actually making a comment to Professor Sharma that you're all, as I said, lovely kids. You're very respectful. You're my impressions are actually very, very positive. In terms of the interaction, maybe you could have been slightly more interactive in places, but also this could be because of, I don't know, being shy or, you know, keeping some sort of distance, I don't know. But overall, my impressions are extremely positive, and I'm really glad I actually had this wonderful experience. And GIAN, I can also see the benefits of such a scheme. It is commendable that the government has such a series of this scheme of actually carrying out thematic workshops, if you could call them. It is great to have the support at the more central level. So this is another comment that I wanted to make, that this is actually indeed a very, very commendable thing, and I applaud it. It's very good. And we're now just delaying things for the inevitable exams later on, yeah. But it is very, nothing to scare you or to worry you, actually.
Can we start?
Of course, of course, of course.
Good morning to all of you. Myself Khushbu Soni, research scholar under the supervision of Dr. Sanjay Singh, CMP College. First of all, I would like to thank the organizers of this event for organizing such a wonderful event for which helps the students in many ways. I'm really grateful to the Dr. Sharma sir, Durgesh sir and their team, and especially to the Professor Fotopoulos. For any event, what is most important is that what we are getting from there. So for me, as I experience foreign delegate, Professor Fotopoulos, and his outstanding work, we learn about that. And what we learn in practical session is that staining of the stress plant with Evans blue and shift staining, and we learn the handling of molecular techniques like RT-PCR and so on. And the most precious thing is that scholars who guided us throughout the session very positively. Thank you so much.
Yes, sir.
Being fortunate enough to have a look.
Actually, I was a bit cold, sir.
No, that's great on your part.
So, good morning to one and all. I am from Shivesh Agricultural Institute from the Department of Genetics and Plant Breeding. I am pursuing my MSc in Seed Science and Technology and the advisor of Dr. Abiotic. So, basically, our work was on seed priming. Yes, sir. So we've been working on seed priming of nanoparticles and even smoke water technique and even plant growth promoting bacteria, sir. So it was a very insightful for sharing your own personal experience and your knowledge, immense knowledge for us. So it has been very helpful for us since we are doing the same work and even meeting new people who are working on the same or in a different way. So it's been very helpful. I wish like everyone to have their thesis very good, and I wish them a very successful way. And thank you, sir. Thank you one and all.
Yes.
No, I'm just saying that you were actually I didn't realize you're a master student. You're actually very proactive, so you have a good one there. Thank you. A very good one. Yes. I thought you were a PhD student actually. Based on your interaction and everything, you were very good.
Definitely, yes.
Good morning, everyone. So I'm Dr. Vani Mishra. I have recently joined Shivesh sir as the women scientist. So at the foremost note, I would like to congratulate the coordinators and the speaker as well for delivering and organizing such a wonderful lecture series. So for us, it was, as all of us said, that we'll agree that it was such an informative lecture. And for me, being a animal scientist, I would say that I've been handling mice models and working on the animal disease pathophysiologies. For me, it was a very insightful lecture. Since I joined with Shivesh sir, so I was a bit nervous also that how to handle all these plant things. But for me, it was just not only the refreshment of the things that I've studied in my graduation or in post-graduation, but also an introduction to what the research is currently being updated in the field of plant stress. So for me, as sir also indicated, Fotopoulos sir also indicated that I pointed out that seed priming is like the thing like vaccination. So for me, it was studying the animal physiology and then coming up to this lecture series and attending and getting information knowledge about the plant physiology. It was for me, it was like correlating the things and coming up to the making up my mind that how can we really relate these things and work on an interdisciplinary network. Because after all these plants and animals we have been studying since inception, that both are connected and depends on each other. So I thank sir again, both Shivesh sir, Durgesh sir and Fotopoulos sir for delivering and organizing such a beautiful lecture series. Thank you so much.
Good morning, everyone. First of all, I would like to congratulate the organizing team for this successful event, and especially for Fotopoulos sir, I would like to mention that I feel blessed that I could interact with you in person, and your words have motivated me so much that I'll not feel demotivated at least in the coming days, and I'll work hard for sure. And your idea of using nosh aspirin in plant science, that was originally a cancer drug, has inspired me a lot, and it is an example of a creative mind. And I will definitely try to incorporate brilliant ideas in my research as well. Thank you so much for inspiring us. Thank you, sir. Good morning, everyone. My name is Harshita and I'm from MNNIT. I am a PhD student. I would like to thank Mr. Fotopoulos sir for this amazing lecture. And I got to learn a lot about biopriming and everything. So I'm really thankful for that. It was a new topic, and then I got to learn a lot of new things related to that. So I'm really thankful for that. And it was amazing meeting you, and you're a really motivating person, and you motivated like all of us. So it was really amazing. So thank you so much, sir.
First of all, a very good morning to all and one present here. Myself Shivani Mehra. This enriching conference under the GIAN initiative centered around the signaling crosstalk during plant abiotic stress phenomena and the transformative potential offered by priming technologies. I stand here humbled by the collective wealth of knowledge shared and gained. Throughout these enlightening sessions, we delve deep into the core concepts of abiotic stress, priming, diverse priming methodologies, and the intriguing crosstalk between the signaling pathways, governing the plant responses under challenging environmental conditions. The hands-on training provided an invaluable opportunity, allowing to witness direct the mechanisms by which priming agents fortify plants' resilience against diverse conditions. Abiotic stress, testing the endurance of plants in the face of environmental adversities. However, in the pursuit of sustainable agricultural practices, researchers and scientists have uncovered fascinating strategies to enhance plant defenses against these stressors. One such strategy gaining increasing attention is priming, as mentioned by Professor Vasileios. Priming. It is a remarkable phenomenon where a plant, when exposed to mild stress conditions, undergoes physiological changes that enable it to better tolerate subsequent, more severe stress. The potential applications are vast and compelling. By harnessing the power of priming technologies, we can imagine a future where crops are equipped to withstand the harshest of environmental adversities, ensuring global food security even in the face of climate unpredictability. Different types of priming technologies such as chemical priming, biopriming, and nanopriming taught us a lot about the upcoming benefits of them. The fine-tuning of priming strategies holds the promise of optimizing resource utilization, reducing dependency on agrochemicals, and fostering an eco-friendlier agricultural landscape. In this journey of fortification, signaling molecules play a pivotal role. These are chemical messengers that facilitate communication within the plant, orchestrating responses to various stimuli. The crosstalk between signaling molecules such as jasmonic acid, salicylic acid, and other secondary messengers amplifies the plant's ability to adapt and resist to abiotic stresses. These complex interactions pave the way for the development of targeted interventions, allowing us to fine-tune plant responses to specific stressors with precision. In experimental endeavors, participants explored the application of staining techniques such as Evans and shifts on seed primed with nano, bio, and hormonal agents like IAA. The following day, we ventured into the basics of RNA isolation procedures, which was shown by a video going in deep into RT-PCR basics, preparing reaction mixtures, and running them on the RT-PCR machine. These hands-on experiences not only deepened the understanding, but also provided practical insights into the application of theoretical knowledge. The prospects in this field are not only significantly compelling, but also holds immense commercial promise. Intellectual property rights, IPR, play a pivotal role in safeguarding innovations and discoveries, such as those explored in this conference. By securing intellectual property associated with novel priming technologies and crosstalk signaling insights, we can foster an environment conducive to investment, collaboration, and technology transfer, as shown us by the Professor Vasileios, sir. This, in turn, can speed the translation of groundbreaking discoveries from laboratories into the fields, benefits from farmers, industries, and the global populace. As we end this gathering, let us carry forth this collaborative spirit and the reservoir of knowledge harvested here. Let us sow the seeds of continued research, innovation, and cooperation, nurturing an ecosystem where discoveries flourish and translate into tangible solutions. I extend my heartfelt gratitude to each one of present here, esteemed speaker, Professor Vasileios, Professor Sir, Dr. G.P. Sahu Sir, Dr. Durgesh Tripathi Sir, Professor Shivesh Sharma Sir, organizers and participants, for your unwavering commitments and passion in exploring the frontiers of science. Thank you.
Anyone else?
In the morning we missed the interacting session, sir. So I just wanted to ask.
Yes, definitely. Go ahead. Please.
So currently I'm doing my MSc, as I said, and we are based on seed planting. So if I want to pursue my PhD degree and even some from here for postdoctoral degree, so can we have insight like how life will be there in Cyprus for students and even any scholarships for Indian students?
Of course. I mean, you should all consider, extending your reach. Doing your masters or your PhDs in India is wonderful, and you should. But I was actually talking to Professor Sharma. At some point, you should all try to get some experience from carrying research abroad. The same way I ask my children to, at some point, do the move and go abroad. If not to carry out like a full postdoctoral project or what have you, even as part of their research. Like most of my PhD students, I organize their projects in a way that they are somehow co-supervised by usually, colleagues of mine in other places that are very respected scientists. Also because this gives them the opportunity to go and spend prolonged periods in the collaborating labs. I mean, I've sent, in my case, people in one of my PhDs, she spent 16 months in Ghent, in Belgium, in a top laboratory. Or now I will send another student to Greece for 4 months. Or there is constant exchange of personnel. So the reason is because you all have to be exposed to other conditions as well. I'm sure that here it is wonderful and highly functional and operational, but it is good to always try to set, broaden your horizons. So by all means, this is my advice. You should all, at some point at least, try to pursue that. Practically speaking, there is a lot of your fellow people, like a lot of people of Indian origin that I am aware of as well, that actually do make the jump to Europe, and actually, seem to be quite happy. I mentioned the example of Jennifer, PhD student that will actually host in my group for half a year in 2024. She is a lovely girl of Indian origin who's actually carrying out a funded PhD project in Poland at present, in a collaborating laboratory. So this is a great example for you to see. In practical terms, it's the same thing as pursuing a postdoc here or a PhD here. It doesn't make any difference. The only difference would just be the unfortunately necessary steps, administrative speaking, of obtaining you would only, as far as I understand, you would only need to go through some steps for the immigration office to because of the fact that it is non-EU member states. For example, for EU people, it is very easy to come over. You don't really need any paperwork, you know. And this is why I very often host people, also because of the ease of the procedure. The only difference with you, for example, making the step would be that you would just follow standard procedures. It's nothing frightening or it's just slightly time consuming. You know, but if you're well organized, that's all there is to it. So keep an eye open. There's positions being advertised all the time, and the same stands for my university and my group. I'm always happy to consider, you know, interest and applications. Of course, always pending availability of funding in projects and so on. You know, so I'm sure everybody's happy. I mean, also colleagues of mine, my department in general is accepting people from abroad very often. So actually invite you to check also specifically my department as well for potential, you know, availabilities. So yeah, you should go for it. Actually, if anything, I encourage you to do it. Thank you. Definitely. It's all about experiences. You have to gain experience, both in science but also in life. And getting exposed to other labs, it's always interesting.
Sir? Okay. So, thank you very much, Vasileios. My pleasure. So, I would like to take this opportunity to thank you once again personally. And during last few days, last 4 or 5 days, I'm sure that all of you, either who joined us in offline mode, in physical mode, or who were there in online mode, and probably they are listening to the interactions and discussions and deliberations we had during last few days. They must have had a very good, wonderful experience as far as learning is concerned and sharing of thought process is concerned and exchange of ideas are concerned. So,
on that particular note, where we had such a wonderful learning of ideas, academic thoughts and deliberations, I must congratulate all of you and personally thank Professor Vasileios for that. And in my opinion, it's one thing to be good at as far as the subject is concerned, which we teach. It's really nice to be great at the subject we taught, and it gives us some satisfaction and confidence as well. But it is great, in my opinion, it's very great and encouraging to be great as far as great educator is concerned, or motivator is concerned, or to be a good human being is concerned. And I find you on the second front, right? So thank you for that, and you have been a great force because I got a chance to be sitting in the sessions. 1 or 2 sessions I sat there, but the feedback I am receiving from the students, both in the WhatsApp group as well as personally, that's really encouraging, and kudos, thumbs up. Thanks are due to Professor Sahu. He was always a one call away. Vasileios, you noticed that yesterday also in the night, in the evening, I just call and he's always there. So as GIAN coordinator, right? As GIAN coordinator, he is the right man for this particular job, right? And person for the job. Yes. This is my 4th course, 3rd with Durgesh, and I find him always raising the standards, right?
He makes it easy.
He makes it easy, right? So solutions are always there, right? So then thanks are also due to our head of the department for her support in organizing and providing the facilities, logistic support, and then most importantly our director. So we had a chance to meet sir in person, and yes, so he always supports and provides all sorts of assistance as far as such type of courses are concerned in the institute.
It was obvious that your director supports innovation.
Yes.
And activities to actually promote excellence.
Yes.
It became very clear yesterday in the meetings.
He sets high standards, because yesterday he pointed out that 2 patents have been granted yesterday also.
Yes,
so we look up to him, right? And then my PhD scholars, right, for all the support, Dr. Vani Mishra, postdoc who has joined, Vikas and his team, SNFC office is his office, yes, all the support. then OSD office, our training and placement office, Department of Biotechnology, EDC staff, all the support, and most importantly the participants. Without you, this course would not have been so fulfilling, and I'm sure that your stay at MNNIT Allahabad in Department of Biotechnology was really fulfilling, right? And you will definitely carry fond memories of this particular course, as well as the learnings and all those things which we interacted. Thank you so very much, and I wish you all the best. And if you require any assistant, our end, Durgesh's end, Sir's end, or Fotopoulos end, just write to us and we'll be very happy to support you in any manner, right? So wish you all the best in your future endeavors. Thank you very much. Sir, you want to speak anything? Okay, right. So on that note, I'll request Durgesh and Professor Sahu. So we'll just be inviting the students and we'll be distributing the certificates, right? कविता एक एक करके यही यहाँ है ना ये सारे खुशबू हाँ हाँ यस फोटो Vasileios. Up, come. Durgesh, please. Give a huge round of applause, right? Saima. She's not here. Megha Baghel. Then Kavita. She's here. वहाँ वो देखो फोटोग्राफ वहाँ आ रहा है कविता शिवम गीता स्तुति दुर्गेश इधर नाउ इट्स फाइन इट्स ओके पद्मजा प्रीति
This is yours.
Ankita. Ankita. Yes. Akriti. Harshita. Dr. Vani.
There are many women scientists are more.
I can see that.
Yeah.
Abhishek. Nidhi.
The newly motivated people. Good.
Shivani. Kiran.
Very highly motivated master students. Yeah. Thank you. Well done. Well done. Congratulations.
Konda Shiva. Manish. और भी डॉक्टर भी तो होंगे ना. Ajay Raj. And Dr. Abhinav.
So, sir is our advisor.
He is supervisor, yes. Right? That's all? Okay. Thank you.
Wonderful.
So we'll join for high tea, right, in the hall. Right. Thank you.
Sir, one feedback everything.
Yes, yes, definitely. हम यहीं पर लें, यहीं ले लें.
We don't fit here, I think.
हाँ.
From all of you, please come once again. Short side वाले, you can assemble it in front. आगे बेटे, one line here.
हाँ, yeah.